Hello, my name is Sara Eglitis, and I'm here at the virtual Roche Discovery Global User Symposium to discuss the application of Discovery AmpHQ kit technology at NovoVita Histopath Laboratory, LLC. On the agenda today, I'm going to start by discussing NovoVita and what we do, then give you some background on the AmpHQ TSA protocol. From there, I'll move on to examples of IHC versus HQ comparison staining, and then share many successful examples of staining using the AmpHQ technology. NovoVita Histopath Laboratory, LLC is a contract research organization, or CRO, based in Boston, Massachusetts, providing a wide variety of histopathology services, as well as automated IHC and automated whole slide imaging. Of that wide variety and full spectrum that we provide, we break it down into 4 separate categories. In the histology section, we do processing, embedding, sectioning, H&E, and special staining. For molecular pathology, there's ISH, FISH, RNA scope, and DNA extraction from FFPE samples. We have a whole span of biosample services, from creating and storing blocks in TMAs, as well as control slides. What I'm really going to be focusing on here today is our IHC capability, where we have automated staining with single and multiplex immunohistochemistry IHC in bright field, as well as immunofluorescence IF. We also do automated image analysis from these stains with whole slide scanned images and high content fluorine and tissue analysis. All of this is available with both human and non-human tissue samples. We have expertise in this whole array of surfaces. Part of our range of state-of-the-art equipment in our facility are 2 members of the Roche Tissue Diagnostics research platform. We have the Discovery XT and the Discovery Ultra. Both of these platforms provide great, reliable, reproducible automated IHC staining, as well as possibility to optimize very specific and specialized staining protocols required by our clients. So now to the fun part, actually talking about the staining itself. When a client comes to us, since we are a CRO, we are based on client projects and not our own. When a client comes to us, we start by optimizing a standard IHC assay. And this assay is as efficient as we can on an Ultra or an XT on the Ventana platform, where you have the primary antibody, either from a vendor or something specialized, and then a secondary antibody, which will then be conjugated with HRP that reacts to the Chromo DAB brown substrate and gives you a single brown color. Ideally, this standard IHC will be able to give you a nice strong stain that is representative of what's happening on the tissue. However, that's not always the case. Sometimes that standard IHC has a lot of background or is very faintly stained and difficult to detect. When that happens, we bring in our AmpHQ kit, the Discovery HQ kit from Roche Tissue Diagnostics, for our IHC with HQ version of the protocol. This is an pyramide signal amplification, or TSA technology, where the HRP activated pyramide binds to tyrosine residues on tissue. Then detection happens with the anti-H reagents and substrate, where we are then able to put on the Chromo DAB stain, the same Chromo DAB that's used in the standard IHC. So you see here on the left, that is the standard IHC that I described in the last slide. And on the the right, it's showing it starts the same way with that primary antibody specific to the project, the secondary antibody with the HRP conjugated to it. But then as we move over even further right, the HRP reacts with the tyramide, deposits the H2 onto tissue, and another antibody comes in that's specifically targeted to that HQ, which is enzymatically linked to another substrate, which will react to the chromomap and deposit the chromogen onto the tissue, giving us an improved signal on a low signal stain or reducing our background. So that's the theory. How do you put it into play? Well, hopefully, since this is a user symposium, you have seen this screen before. This is our Create New Protocol screen on the Discovery platforms. The one I'm showing you here is for the Discovery RUO. And hitting the Create Edit Protocol button brings you this blank screen. 3 checkboxes are available: the first sequence, the counter stage, and the side cleaning. When you're setting up a protocol, you're going to start with that first sequence. By clicking that button, you'll open up a full range of protocol variables that you're able to insert into your IHC staining protocol. Things like baking and deparaffinization and inhibiting endogenous peroxidases, as well as the primary antibody application and incubation time. That's something you need to figure out, whether it's a standard IHC or an AmpHQ IHC assay. What I'm focusing on today and what I just showed you in the previous slides comes after that primary antibody. First off is this little box called Multimer HRP. This is your secondary antibody that comes in conjugated with HRP. When you click the box, it'll give you options to select which Multimer HRP reagent you wish to use. There are OmniMap and UltraMap options. And what you really need to look for here is what species your primary antibody is. Is it a rabbit? Is it a mouse? And then select the appropriate one from the box as shown on the right. From here, you can then go to the DAB and counter state, and that's how you create a standard IHC assay protocol. However, if you need that extra boost, that amplification, you're going to move to a checkbox a couple more below, and that's the Discovery Amplification selection. The Discovery Amplification comes as a kit, and you have a couple dispensers in it. You're going to have the Discovery Amp TSA HQ and the Discovery Amplification H02HQ, where you need to set the incubation time. And then you also need to decide which Discovery Multimer to use. As you've heard me say here, we're using the HQ version. So of the 3 that I have available as options on the right, the anti-BF is not the one we're using here. The 2 HQ options are AP and HRP, and which one you choose depends on what chromogen you're going to use for your final assay. The most commonly used one is the DAB, which is often from the chromomap kit, very reliable, very commonly used on the Ventana line. There are some needs for an AP detection when you're not using a chromomap DAB, or, as you'll see soon, a range of non-DAB chromogens available from the Ventana line that are HRP activated. So here's a great example of what moving from an IHC, standard IHC protocol to an AmpHQ protocol looks like. On the right, we have the first run of this immune cell antibody staining on syngeneic mouse spleen project. With that standard IHC protocol, we get a weak signal. We can see that there are positive cells, but they're not popping in the way we really expected to be able to analyze these immune cells. So we added an amplification step. We did that AmpHQ, and we also realized that the end game of this particular assay was going to be multiplexing. And when you multiplex, you're adding more than one antibody onto the tissue on a single slide. And you need to use different colors for the different antibodies. For this particular target, we're not going to be keeping the brown. We decided to switch to a purple. So in the process of adding the amplified signal with the AmpHQ kit, we also switch our chromogen from DAB with the Chromo kit to the Discovery Purple HRP, which is what you see here on the left, beautifully showing distinct purple immune cells in a field of the counterstained hematoxylin nuclei. Here's another example of some of our standard optimization sequences. This happens to be a tumor cell antigen staining on human endometrial carcinoma. In the middle, you'll see our initial pass with the optimization, the standard IHC. We have some very weak brown staining on the membrane of the tumor cells. And if we had continued with this, we feared we'd get a lot of false negatives in the project. So we decided to add that amplification step. And by keeping the dilution of that primary antibody consistent and simply adding in that HQ amplification step, we were able to get distinct brown staining that didn't look very faded. You could really visualize what the tumor cells were for expression levels. Because we are scientists, we do make sure to run our negative controls, and that's what you see all the way on the right. This is our rabbit IgG, running negative controls with IHC assays. Make sure that you use an IgG that's a consistent species with the species of your primary antibody, in this case, rabbit, at the same concentration using the amplification HQ kit, and we see a nice negative stain compared to our AmpHQ staining all the way on the left. However, you may be able to notice, depending on how your screen is, that there is a faint brown blush in our rabbit IgG, just based on the tissue itself. This did not concern us because this was our HQ version, and we could easily read around that area and see what the real staining was. However, if this was a negative control to the middle panel, our standard IHC, much of what would have been considered positive with that IHC would have been the same as our negative control, meaning that our fear of false negatives was very valid, and moving to the AmpHQ was the right move for this particular project. We are also able to use this technology in more cell-based staining, not simply tumor tissues or for immune cells within the tissue. Here's a PBMC sample with a phosphorylated antibody. And I'm sure many of you in the crowd have dealt with the trials and tribulations of dealing with phosphorylated antibodies. They're not the easiest ones to deal with. And with our standard IHC, we have really weak signal. However, putting in that AmpHQ sequence, we have a very strong, distinct brown stain. We're able to detect which of these PBMCs are positive and which are negative. Here's another great example of not needing to use the DAB to have distinct IHC staining using the Ventana and when using the AmpHQ kit. Here's strong signal detection with purple on a non-small cell carcinoma sample. We're able to see distinct membrane staining around the tumor cells, differentiated from your blue hematoxylin nuclei. I have heard some questions about the nearness in color between a purple hematoxylin in a purple color when you choose your chromogen. So it's definitely something worth considering as you put your protocol together, which hematoxylin you're using, how blue it can be, how purple it can be, and the amount of bluing time that you use. I believe there are 2 different hematoxylins that Ventana provides, hematoxylin 1 and hematoxylin 2, and each lab has their own preference. So be sure when you're switching from a brown to a different color to really keep that in mind of what your counter stain will look like. Now I'm going to talk about different types of projects we've seen and use the AmpHQ kit for. For this one project, the client came to us with a batch of archived slides or blocks. Not quite sure. It could be slides or blocks. Either way, they were very old FFPE samples, and they were poorly preserved by the time we got our hands on them. We started with the traditional IHC, our standard IHC, and there was no signal for the target we were interested in, but there was a lot of background staining. This is the one on the left where we have that nuclear stain, a little bit of a blush in between. We weren't going to be able to detect what we knew was going to be a small, low amount of signal antigen. We moved to the Discovery AmpHQ kit, which helped us minimize that background staining, remove that nuclear staining, and be able to clearly identify that low-level positive antigen staining, as you can see in the highlighted box on the right. Another type of study that we've done for a client is for detecting drug cross-reactivity in normal and target tissue. In this project, the client gave us different batches of samples, some of which had been exposed to the drug, some which had not, some which were tumor, and some which were normal organs. And we were to confirm if the antigen of interest was present in which batches of sample, hoping, of course, that the antigen of interest would only in the targeted tissue exposed to the drug. Here are 2 great examples of what came out of that project. On the left is a sample that was completely negative and had never been exposed to the drug. On the right is a sample that was exposed to the drug and displays drug-specific binding. And the assay that we used for this binding was the TSA Technology with the AmpHQ. So looking at a single stain is very useful, but sometimes you need a little bit more context from a single slide. And in that case, you're going to use a multiplex protocol. Using the Ventana system, you're able to sequentially add additional antibodies after that first sequence. And where you're going to be doing that is in that same Create Edit Protocol screen that I showed you earlier. At the end of the first sequence, right before your counter stain, you select dual sequence. You can keep going down and down, triple, quadruple. I believe at the last update, there's 7 or 8 you're able to add in sequence in that way. So here's an example where we went to a dual stain from our single. And one of the most efficient ways to do that is by keeping your staining from a single optimized assay. In this case, ours on the CD8, we're keeping it with that ChromoMap DAV brown stain and simply adding the optimized protocol for our CD19, but switching from the DAV to the teal. The teal is also HRP activated as a chromogen, so you don't have to change any of the linking antibodies above it for it to give this great robust stain. Here's another example of the same idea. We start off with Nectin-4 single optimized assay with a brown DAV stain using that AmpHQ kit, and wanted to see another antibody on the same slide. In this case, instead of the teal, it's a purple. Purple is also an HRP activated chromogen, which has very robust, reliable performance, and is my second choice. Might be the same level as teal. They're my two most used non-brown chromogens as I start to create complex protocols. Multiplexing, of course, is limited to bright field and chromogens, and neither is using this AmpHQ technology to really boost your staining intensity. We've had success with using it in immunofluorescent assays as well. Here's a great tumor example where we're looking at CD4 in green, CD8 in red, and we get great cellular staining in this tumor sample. Moving from 2-color multiplex to 3, this is a great example of a project that came from a client interested in 3 separate tumor-specific antigens, which they knew were prominent in this type of tissue. However, they were interested in what the distribution actually is. Are all 3 antigens on the same cells? Do they have patches within the tumor? And about that really makes you think of how to construct this type of protocol, where you're able to look at each antigen separately and also any co-expression. So the colors we chose for this protocol were teal, purple, and yellow. That's because they're very distinct colors individually, and helpfully, they allow for a new color when they are co-expressed. So the teal and the purple co-expressed on a cell give this rich dark blue, which you're seeing here throughout the tissue, as there are very few cells which are strictly purple or strictly teal. Get that rich blue color. When purple and yellow are co-expressed, they give an orange, which we have 1 or 2 cells here, not that many. In this particular project, we were able to explain for this sample that the antibody 1 and antibody 2 can co-express, while antibody 3, that yellow, be on different cells. And if yellow and teal were together, it would give you this green cell. Keeping that co-expression in mind and adding more colors, here's an example of looking at a tumor microenvironment where we have the same cell types that we're looking at. We're not actually interested in a co-expression. We want to be able to see each color distinctly. So the CD4 is in brown, which will not do a co-expression color with the CD8 in purple. CD8 and CD19 are different cell types, so they're not going to be co-expressed. And the blue is a monocyte, which I've highlighted here because they are a little bit difficult to find if you're not looking for them. They're all distinct separate colors. So sometimes when you're multiplexing with this AmpHQ, you need that co-expression, like in the previous slide, and sometimes you need them distinctly separate, like in this slide. And you have to pick your chromogens accordingly. Looking at slides one by one is a very helpful way to get quick context about that particular slide. But we get a lot of projects through here that need bigger picture answers than just a one-by-one, slide-by-slide type of description. And for that, we move on to TMAs. We had a project recently where it was a low-expressed antigen on a panel of tumor types, and this client was interested in knowing what the patterns are for their particular subtype that had high expression versus low expression. And we were able to do that by using an AmpHQ to really detect that low signal antigen, and then use our automated full slide imaging and image analysis to create a TMA map, screening for indications and evaluating the incidence of diseases in those diseases of interest for the client. Another piece of our state-of-the-art capabilities is high-quality 100x oil whole slide imaging, giving us up nice and close cellular views of the tissue. We are sometimes using our AmpHQ kit technology when staining these samples. And at the 100x oil, you're still getting nice crisp stain. It doesn't become fuzzy or indistinct. We still get that nice crisp On top of that, a nice 100x oil allows for Z-stacking images, where you get a better three-dimensional feel of how the cells are in space on that tissue. As you can see here on the left, we have that nice crisp cellular look, and on the right, still a very pretty 100x image, but without that added context that a Z-stacking allows. So in summary, I've presented on the application of the Discovery AmpHQ technology at NovoVita. NovoVita Histopath Laboratory is a CRO located in Boston, Massachusetts, specializing in automated histopathology services, including high-quality immunohistochemistry. The Discovery AmpHQ kit provides sensitivity of IHC assays compared to the standard IHC assays, which helps to reduce background and improve signal. The Discovery AmpHQ kit protocols can be used for a variety of project types, including but not limited to the ones I've described here, such as looking at tumor antigens, reducing backgrounds on old, poorly preserved samples for use in TMAs in both single-stain and multiplex assays. Before I wrap up, I would give a huge shout out of thanks to the Novo Histopath team. Helen, Andrew, Chris, Julian, Selena, and Justin were a big help with putting this presentation together. And finally, I've been Sara Eglitis. I hope you learned how to use the AmpHQ kit and the benefits it can provide to your IHC staining. Thank you very much for your attention, and I hope you enjoyed the Roche Discovery Global User Symposium.